Bismillahirrahmanirrahim. Let's discuss the morphology of cell and tissue injury. We are having two types of cell, that is reversible injured cell and irreversible injured cell. मॉर्फोलॉजी जानना बहुत ज़्यादा ज़रूरी है क्योंकि कुछ सेल होते हैं जिनकी डेथ हमारे पास बहुत ज़्यादा जल्दी हो जाती है और कुछ की बाद में होती है. इवन सेलुलर फंक्शंस जो हमारे पास होते हैं वो पहले लॉस्ट हो जाते हैं सेल के, जबकि हमारे पास मॉर्फोलॉजिकल चेंजेस जो हमारे पास होती हैं वो हमारे पास बाद में होती हैं. मायोकार्डियल सेल है, हमारे पास जैसे ही इस्कीमिया होता है 1 से 2 मिनट के अंदर नॉन कंट्रैक्टाइल हो जाता है, जबकि हमारे पास डेथ तब तक नहीं होती है जब तक कि 20 to 30 मिनट्स नहीं गुजर जाते हैं. तो इससे हमें पता चलता है कि मॉर्फोलॉजिकल चेंजेस और सेलुलर फंक्शंस का आपस में क्या रिलेशन है. दो तरह के सेल होते हैं, वन इज कॉल्ड एज रिवर्सिबल इंजर्ड सेल, अनदर वन इज कॉल्ड इरिवर्सिबल इंजर्ड सेल. रिवर्सिबल इंजर्ड सेल इरिवर्सिबल इंजर्ड सेल में तब कन्वर्ट होता है जब हमारे पास दो कंडीशंस होती हैं. Either there is inability to correct the mitochondrial dysfunction, that is the lack of oxidative phosphorylation and ATP generation, or there is profound disturbance in membrane function. यहाँ में देखते हैं हमारे पास रिवर्सिबल सेल इंजरी और इरिवर्सिबल सेल इंजरी है. दिस येलो इज फॉर रिवर्सिबल सेल इंजरी. सेल फंक्शन टाइम के साथ साथ आप लोग देख सकते हैं कि हमारे पास क्या होता जा रहा है, कम होता जा रहा है. दिस इज द फैक्ट एंड और हियर इट इज ड्यूरेशन ऑफ इंजरी. जबकि सेल डेथ हमारे पास जो होती है इरिवर्सिबल सेल इंजरी के अंदर क्या होता है, सेल हमारे पास सेल डेथ की तरफ जो होता है वो प्रोग्रेस करके जाता है. अल्ट्रा स्ट्रक्चरल चेंजेस जो हमारे पास होती हैं वो भी इरिवर्सिबल सेल इंजरी के अंदर ज़्यादा होती हैं. लाइट माइक्रोस्कोपिक चेंजेस भी ग्राफ हमारे पास इंक्रीज होता है इरिवर्सिबल के अंदर, और मॉर्फोलॉजिकल चेंजेस का ग्राफ भी इरिवर्सिबल सेल इंजरी के अंदर ज़्यादा होता है. और इरिवर्सिबल इंजर्ड सेल दो टाइप के होते हैं, नेक्रोसिस और एपोप्टोसिस. लेट्स डिस्कस रिवर्सिबल इंजर्ड सेल. इट शोज हाइड्रोफोबिक चेंज और सेलुलर स्वेलिंग और what is known as vacuolar degeneration. जबकि फैटी चेंज हमारे पास रिवर्सिबल इंजर्ड सेल के अंदर होता है. अब पहले डिस्कस करते हैं हाइड्रोफोबिक चेंज, सेलुलर स्वेलिंग एंड वैक्यूलर डिजेनरेशन को. एक बात पता हो, हम रिवर्सिबल जो हमारे पास सेल है इंजर्ड सेल, इनको हम डिटेक्ट कर सकते हैं बाय फ्लो साइटोमेट्री एंड सेल्फ मॉर्फोमेट्री. दे आर द न्यू टेक्निक्स यूज्ड टू डिटेक्ट नॉर्मल एंड एबनॉर्मल सेल्स. How it is repaired? Nuclear damage can be repaired by nucleotide repair mechanism or by base mechanism. Changes in cellular swelling, that is included in vacuolar degeneration or pallor, increased turgor, increase in weight of the organ, and microscopic examination may reveal small clear vacuoles within the cytoplasm. These actually represent the distended, or you may say the pinched off segment of the endoplasmic reticulum. Fatty change common site is liver. Also occur in skeletal muscle, heart and kidney. Causes include alcohol, diabetes mellitus, obesity, pregnancy, protein energy malnutrition. Non-alcoholic fatty liver is also the cause of fatty change. Fatty change is manifested by the appearance of lipid vacuoles in the cytoplasm. It is principally encountered in cells participating in fat metabolism. For example, hepatocyte myocardial cell, and is reversible. An injured cell show increased eosinophilic staining that is more pronounced and progress to necrosis. Intracellular changes include the plasma membrane alteration, mitochondrial changes, dilation of endoplasmic reticulum, and then there will be detachment of ribosomes along with it and dissociation of polysomes. Then there is nuclear alteration. So the nucleus will show the clumping of chromatin, and cytoplasm may contain phospholipid masses, which is called as myelin figure, which are also derived from damaged cellular membranes. Okay, let's move toward irreversible injured cell. The first one is necrosis. Type of cell death associated with loss of membrane integrity and leakage of cellular contents, culminating in dissolution of cell, largely resulting from degradative action of enzymes on lethally injured cell. Necrosis क्या है, सबसे पहले पता होना चाहिए. Irreversible injured cell होते हैं, सेल डेथ होती है. टाइप ऑफ सेल डेथ एसोसिएटेड विद लॉस ऑफ मेम्ब्रेन, मेम्ब्रेन इंटीग्रिटी लॉस हो जाती है. जबकि जब मेम्ब्रेन इंटीग्रिटी लॉस हो जाती है तो जो सेल के अंदर सेलुलर कंपोनेंट्स होते हैं वो हमारे पास लीक हो जाते हैं, सेल हमारे पास डिसोल्यूट हो जाता है और सेल डिग्रेड हो जाता है. जब हमारे पास लीकेज होती है ना सेलुलर कंटेंट्स की, तो पता होना चाहिए कि लीकेज की वजह से हमारे पास क्या होगा, लीकेज ऑफ सेलुलर कंटेंट्स की वजह से हमारे पास इन्फ्लेमेशन होगी और जो एंजाइम्स का एक्शन होता है, these enzyme can be derived from lysosome, that is dying cells or leukocytes. तो सेल हमारे पास सेल डेथ होगी, लीकेज होगी सेलुलर कंटेंट्स की, that will result in inflammation. And over here the degradative action of enzyme means that these enzymes can be derived from lysosomes, either it is dying cell or it can be leukocytes. Hope definition is clear. Mechanism of necrosis दो तरह से, either it would be denaturation of proteins or there would be enzymatic degradation. ठीक है, enzymatic degradation can be autolysis, heterolysis, depends. मॉर्फोलॉजी की बात करें, 3 चेंजेस हैं, या साइटोप्लाज्मिक चेंजेस होंगी, न्यूक्लियस के अंदर होंगी, एंड फाइनली वी विल डिस्कस द फेट ऑफ नेक्रोटिक सेल कि फाइनली फिर क्या होता है नेक्रोटिक सेल के साथ. साइटोप्लाज्मिक चेंजेस इंक्लूड इंक्रीज eosinophilia. That is, there would be increased binding of eosin to denatured cytoplasmic proteins and loss of basophilia. This basophilia normally is imparted by the ribonucleic acid in the cytoplasm. Cell को क्या होगा? Cell will have a glossy, homogeneous appearance. Mitotic figures are more prominent. और इस सब के बाद जब आप सेल को देखते हो, तो when enzymes have digested during cytoplasmic organelle, or you observe the cytoplasm, it become vacuolated and appear like moth-eaten cytoplasm. So the very important point is that while discussing the necrosis, you should remember that its cytoplasmic changes is like moth-eaten cytoplasmic appearance. There will be discontinuous membrane and dilation of membrane disrupted lysosomes. Along with that, there will be intracytoplasmic myelin figures disruption and appearance of large amorphous densities. Nuclear changes include karyolysis. Karyolysis क्या चीज़ होती है? It is faded chromatin basophilia. Pycnosis क्या होता है? It is nuclear shrinkage, increased basophilia, DNA condenses and shrunkens. And karyorexis means pycnotic nucleus that undergo fragmentation. Fate of necrotic cell may persist, are digested, or disappear. Dead cells may be replaced by myelin figures. या तो हमारे पास phagocytosis होने लग जाएंगे, या हमारे पास डिग्रेड होने लग जाएंगे किस चीज़ के अंदर? फैटी एसिड के अंदर. So necrotic cell ने क्या करना है? Necrotic cell जो हमारे पास dead cell है, that would be replaced by myelin figure. These myelin figure either would be phagocytosed or they would be degraded into fatty acid. These fatty acid will bind the calcium salts, which may result in the dead cell ultimately becoming calcified. Let's talk about the pattern of tissue necrosis. The first one is coagulative necrosis. Coagulative necrosis, underlying tissue architecture is preserved. Injury denatures structure and proteins, enzymes. Block the proteolysis of dead cells. Eosinophilic anucleated cell persist. Recruitment of leukocyte. Phagocytosis to remove cellular debris. For example, infarcts in solid organ except brain. Basically, coagulative necrosis is that form of necrosis in which the underlying tissue architecture is preserved for at least several days. And it occurs in solid organs. Liquefactive necrosis is seen in focal bacterial fungal infections. Because microbes will stimulate the accumulation of inflammatory cells. Enzymes, what they do, they will digest the tissue. So as a result, hypoxic death of cells within the central nervous system, it is seen that they also evoke the liquefactive necrosis. So what happened? That whatever the pathogenesis, the dead cells are completely digested. Once they are digested, they transform the tissue into a liquid viscous mass, and eventually that is later removed by phagocytosis. Initiated by acute inflammation, as in bacterial infection, the material is frequently creamy yellow called as pus. Caseous necrosis. Caseous means cheese-like. It is tribal, white appearance. Collection of fragmented cell is seen, and complete obliteration of tissue architecture occur. It is mostly present in foci of tuberculosis infection. Coagulative necrosis और caseous necrosis में important बात ये है कि over here in coagulative necrosis the first point में लिखा है कि tissue architecture क्या होता है? preserve होता है. जबकि caseous necrosis में यहाँ पे सीधा सीधा लिखा है कि complete obliteration of tissue architecture is not preserved. तो वहाँ पे preserve है tissue का और यहाँ पे preserve नहीं है. यहाँ पे ग्रेनुलोमा की फॉर्मेशन होती है. व्हाट डू यू मीन बाय ग्रेनुलोमा? फर्स्ट करें कि मैंने कहा कैशियस नेक्रोसिस में देयर इज अ येलोइश वाइट अपीयरेंस. सो कैशियस नेक्रोसिस में येलोइश वाइट अपीयरेंस हो गया. इट इज सराउंडेड बाय इन्फ्लेमेटरी सेल्स. So this is called as granuloma. How do you define granuloma? The area of caseous necrosis is often enclosed with a distinctive inflammatory border, and this appearance is a characteristic of a focus of inflammation known as granuloma. Fat necrosis is of two types, enzymatic and traumatic. Enzymatic is followed by acute pancreatitis, release of lipases that causes dissolution of fatty acid, so saponification occur. And traumatic is more common in female breast, no enzyme release, trauma to fatty tissues. Fibrinite necrosis is hallmark of autoimmune disease and hypertension. In it, immune complex plus fibrin is present, and it combined to form the fibrinite. The deposited immune complex together with fibrin that has leaked out of the vessel produce a bright pink and amorphous appearance that is called as fibrinite, and the immunological mediated disease, that is polyarthritis nodosa, is the example in which this type of necrosis is seen. Let's talk about another form of necrosis, that is gangrene. It is defined as a form of necrosis with superadded putrefaction. Let's discuss its three different types: dry gangrene, wet gangrene, gas gangrene. Side dry gangrene commonly occurs in limb. Wet gangrene occurs in bowel. Mechanism arterial occlusion. Wet gangrene more commonly venous obstruction, less commonly arterial occlusion. Microscopy, organ is dry, shrunken and black. In wet gangrene, it is moist, soft, with swollen, rotten and dark. Putrefaction is limited due to very little blood supply, while wet gangrene is marked due to stuffing of blood with blood. Organ with blood means that organ is filled with blood, so it appears the wet. Along with that, it mostly occurs in venous obstruction, or in bowel, or in intestine. So basically, wet gangrene is a case of surgical emergency. Line of demarcation is present at the junction between the healthy and gangrenous part, while in wet necrosis there is no clear line of demarcation. Bacteria fail to survive, and in wet gangrene numerous present. Prognosis better due to little septicemia, and it is bad due to toxicemia. Causes are gut disease, Raynaud disease, arteriosclerosis, while in wet gangrene it occurs mostly in intestinal diseases. Let's talk about the gas gangrene. Definition: it is infection involved in injury or surgical wound following infection by Clostridium perfringens. Morphology: in gas gangrene, you would see an area that is gross, it is swollen, painful, edematous, cryptant, dark black, pigmentation, and foul smelling. That's all. In the next video, we are going to talk about the apoptosis, necrosis and apoptosis, and autophagy.